fact that I am here, you probably realize that this is a physiology lecture. I hope it has reached you through websites or whatever that there was a change in the anatomy. Hasn't? Well, it's certainly on the department, our departmental site, it's up there that it's a physiology lecture at this time. In the program. Oh, okay. Excuse me? Only today, yes. This is, the anatomy department couldn't do something, so they asked me to come and do this lecture. Okay, well, anyway, in my last lecture, we have discussed the neuromuscular junction as an example for a chemical synapse. Today, I will continue with showing you a number of different synapses and a number of different interactions in the nervous system, as well as receptor function. Before I do that, I would like to say that from notes or discussing with students, I got the impression that the website has the old lecture notes, so I told them to exchange them, so please look up to the lecture. The new notes should be up, I hope. Well, anyway, the things are not that different from last year, it's just that I made some drawings. Okay. So, obviously, last time when we discussed the neuromuscular junction, I said that in the neuromuscular junction you have a motor neuron and the skeletal muscle fiber. And this motor neuron stimulates the skeletal muscle fiber through the neuromuscular junction. This system we refer to as the somatomotor system, and as discussed, this synapse is a neuromuscular junction, and in this synapse, the neurotransmitter is acetylcholine, and this neurotransmitter acts on a nicotinic type acetylcholine receptors. This is what we discussed in very detail, but this is the scheme and drawing type that I will be using, describing you two other type of interaction, which is found in the autonomic nervous system, simply because in your studies from now on, the autonomic nervous system and the regulation through the autonomic nervous system will be one of the most important ways of regulation that we will see. So, in the autonomic nervous system, you can divide into two different parts, the so-called parasympathetic and sympathetic nerves. The difference, as you see, is that in case of the autonomic nervous system, both parasympathetic and sympathetic, we will have two neurons and a cell where the autonomic nervous system acts. This is what you find outside of the central nervous system. Each and every case. So you have an effector cell. In case of the parasympathetic nervous system, if this vagal nerve might say something, vagal nerve belongs to the parasympathetic nervous system. So effector cells will be smooth muscle cells. This I will be discussing in a few lectures from now. Cardiac muscle will be a big topic in the next block of lectures, and glands will be topics as things go on. So obviously there are two neurons and two synapses involved. The first synapse along the way, this would up here would be the central nervous system, is so-called the synapse at the parasympathetic ganglion, and the second the neuron that comes is the so-called preganglionic neuron, which comes to this synapse. And then from the ganglion, a parasympathetic postganglionic neuron goes on. If one looks at the synapse, then what you would have to know, and please understand, these are things that kill people if you do it wrong. And these are things that will fail you in an exam if you say it wrong. In this synapse between two neurons in the parasympathetic nervous system, the neurotransmitter is again acetylcholine. It acts again on nicotinic acetylcholine receptors, but these differ a little bit from those seen at the muscle. I will come back to the difference in a second. 2-3 slides from now. In case of the postganglionic neuron, here where the nervous system sends the information on to the cell where it wants to make an effect, the neurotransmitter is again acetylcholine. However, it acts on muscarinic cholinergic receptor. What does that mean? I will come back to it and explain. In the sympathetic nervous system, the most common arrangement looks exactly like that in the parasympathetic system. You have an effector cell, again, smooth muscle, cardiac muscle, gland, and you have two synapses from the fact that you have a sympathetic ganglion into which a preganglionic neuron goes, from which a postganglionic neuron starts. Okay? And again, please, again, this is a must. The neurotransmitter here in the sympathetic ganglion, that is between two neurons here, is again acetylcholine with nicotinic acetylcholine receptors of the neuronal subtype. So this is the same for the two classes of the autonomic nervous system. However, down here, that is where the sympathetic nervous system acts on a cell, the neurotransmitter is norepinephrine or noradrenaline. Those liking Greek better, please use noradrenaline. Those liking Latin better, you can use norepinephrine. Adrenaline and epinephrine are word-to-word translations for on the kidney. Simply referring to the fact that these guys can come from the adrenal gland. Okay, norepinephrine acts on adrenergic receptors. What those are, I will come back to that in a second. So, nicotinic acetylcholine receptors, I would like to go through fast. These are ionotropic receptors as discussed with the 5 subunits, non-specific cation channels, and they do a membrane depolarization with 2 subtypes, a neuronal subtype which has a subunit composition of such. In this case, the agonist is acetylcholine and nicotine, of course, with an antagonist of hexamethonium. Oops, sorry. Please recall that the muscle subtype had a different subunit composition. Ladies and gentlemen, you do not need to know the subunit composition. I'm just showing you this is the difference. But please note that you do need to know the antagonist of both the neuron and the muscle subtype. For example, last time I showed you that D-tubocurarin would be an antagonist for the muscle subtype. It is absolutely essential if you want to block this neuronal transmission and you give D2-glucuronide, the person will die. So these things are not to be mixed. Muscarinic acetylcholine receptors are, on the other hand, metabotropic receptors. They are linked to G proteins. I hope metabotropic receptor and G protein does mean something to everyone. Yes, first year and so on. G proteins and okay. For these receptors, they got the name from the fact that there is a mushroom, the fly agaric or Amanita muscaria, which produces a substance called muscarin, which is the agonist for this particular receptor. Please recall, nicotinic receptor, nicotine was the agonist, muscarinic receptor, muscarin is the agonist. Very simple to remember. The mushroom has been in use a couple of thousand years ago when people of tribal, how should I say, magicians, put it into their mixture, eat it, and that produced hallucinations, and that's how they communicated with the other world. Please, ladies and gentlemen, don't try to make this cocktail. It can be dangerous. As an antagonist, please remember the name atropine. Atropine comes from the plant called Atropa belladonna. Can someone translate belladonna for me? Belladonna. Beautiful mist. What do you think? Why would a plant like that be called as beautiful mist? Yes, the atropine acting on these muscarinic receptors causes the dilation of the pupils. And people in the Mediterranean, Spanish areas, and they thought that if a lady has wide pupils, that's beautiful. So ladies did this in the Middle Ages. So please remember the name muscarin as the agonist of these metabotropic receptors, and atropine as an antagonist of these receptors. There are a number of different subtypes of these receptors. We will not go into these details in second year, but pharmacology will talk you into all. Most of them act, or sorry, 3 of them actually, the odd numbers act on IP3, GQ proteins, if that means something, whereas the even ones act on GI proteins, decreasing cyclic AMP. Adrenergic receptors are subdivided into 2 groups. Beta adrenergic receptors, they are coupled again to G proteins and result in the increase of cyclic AMP. Actually, all beta adrenergic receptors do. There are 3 different subtypes. I didn't want to go into detail. They act on, for example, your heart. So when you are, this is a sympathetic activation, so when you are frightened, when you are stressed and so on, your heart starts to go faster. All that is this, what we are discussing. So this you will learn when you get to the cardiac function. And on these receptors, epinephrine is more effective than norepinephrine. The other group of receptors are called alpha adrenergic receptors. Alpha-1 adrenergic receptors are coupled to G proteins and IP3, whereas alpha-2 receptors are coupled to GI proteins and decreasing cyclic AMP. In this case, norepinephrine is more effective than epinephrine. This is the basics that all second-year physiology students need to know. They came from deep sea, like that. Everything else, at the moment in Hungary, the number of medications that somehow act on one of those receptors that I have just listed exceeds 100. That is probably true for all countries in the world. Maybe you have heard the expression beta blocker. Everything that you take for your heart and so on, blood pressure, all this. So when you get to pharmacology, you will learn approximately 40 different substances that act on these receptors. So in second year, we will stop with, I don't know, I said I think 5 or 6. But these you must know. Okay, so how does a cholinergic synapse then work? We have discussed the events on the postsynaptic cell, the nicotinic acetylcholine receptor. Obviously, the neurotransmitter is acetylcholine. What I would like to emphasize at this point are the following things. First, look at the vesicle, and please note that there is not only acetylcholine in the vesicle, but in this case, ATP and some prostaglandins. This is true for most synaptic transmission, that next to the basic neurotransmitter, other substances might be found in the vesicle and co-released with the neurotransmitter and act on the postsynaptic cell. Secondly, I mentioned that in the synaptic cleft there is an enzyme called acetylcholinesterase, and this acetylcholinesterase cleaves acetylcholine to choline and acetate. Acetate simply diffuses out from the synaptic cleft, whereas choline is being taken up back into this presynaptic cell via a co-transport with sodium. This choline with the acetyl coenzyme A is being used to produce the acetylcholine. The enzyme is choline acetyltransferase. You know, biochemists like to name things of what happens. So choline acetyltransferase is what puts the acetate on the choline to make acetylcholine. This is then put into the vesicle, and this vesicle is then ready for release again. Please also note the fact that if this would be the case, this membrane would always grow and grow. So obviously, membranes patches are taken back in and reused for making new vesicles. These vesicles are transported back and forth along the axon. How does an adrenergic synapse work? Well, as I mentioned, in the adrenergic synapse, the neurotransmitter is norepinephrine. There is no enzyme in the synaptic cleft, that would cleave norepinephrine. So two things can happen. Norepinephrine can simply diffuse out from this synapse, or can be taken back into the presynaptic cell. This is again a co-transport, and norepinephrine sodium chloride co-transport, which has a blocker that for some reason is known to everyone. Why that is the case, I don't know, but for some reason I find that all students happen to know this specific blocker. It's called cocaine. Ladies and gentlemen, the effects of cocaine are also known, for some reason, to students. If I would have to ask, does anyone know muscarine? Probably none of you would have known. For some reason, cocaine, everyone knows. Even the effects are known. Please note that although the effects are known, it might be that you didn't know so far why cocaine has the effects that it does. The reason is that all adrenergic and related substances, including dopamine and serotonin, synapses, so where norepinephrine, epinephrine, dopamine, and serotonin are the neurotransmitters, this particular transporter is present to pick up the neurotransmitter. And cocaine stops this from working. That is, it prolongs the presence of the neurotransmitter in the synaptic cleft. And this is why it has the effects that it has. Norepinephrine then can be either cleaved inside the cell. This can happen in the presynaptic cell. There can sometimes be enzymes in the postsynaptic cell. Norepinephrine that diffuses away is cleaved in the, degraded in the liver. And the enzymes involved, some former Chinese students told me that these are very conservative enzymes because they are called Mao and Comte. But before you misunderstand, Mao stands for monoamine oxidase and not Mao Zedong. And Comte has nothing to do with philosophy, it's catechol methyltransferase. So the two enzymes that cleave the norepinephrine are these two enzymes, or norepinephrine can be transported back into the vesicle, and there is again a transporter here which is blocked by reserpine. Interestingly, this transport is again a norepinephrine transport. Nevertheless, reserpine is probably not known to anyone. That is because they although they are both norepinephrine transport blockers, they have partially antagonistic effects, since if reserpine is applied, then these vesicles tend to have less norepinephrine, therefore serotonin or dopamine, therefore the amount of norepinephrine released becomes less, and that is not that will not cause the effects what you see when there is more norepinephrine out here. So this is the amount of pharmacology that you need to know in order to understand the things that will be said when we get to the organ physiology. Moving along, I would like to say a few words about synaptic integration and make some things clear in terms of why it is important for everyone to understand electrotonic potentials. Action potentials, everyone, we have to know action potentials because they code everything. Well, please understand that electrotonic potentials are just as important, and that is because if you take a neuron, this is of course a very crude representation of the neuron, and if this would be the soma of a motor neuron, then the axon that starts here in this scale would end somewhere at the train station. The dendritic branch that you see here is again a crude underrepresentation of the neuron. If this would be the neuron, then this dendritic tree would fill this entire building. Of one neuron. If you would blow up the neuron to have this size of a soma, then the dendritic tree would fill the building. And the axon would go down somewhere to the train station. That's That is what we will be discussing in a second. Before I go, I would like to emphasize the fact that it is always good if a teacher says big numbers. So I will say big numbers. You have probably heard that there are so many neuronal cells in our neurons in our central nervous system. They estimate it to be 10 to the 10, 10 to the 11. Now each and every neuron has approximately 1,000 synapses on its dendritic tree. Motoneurons have more than 2,000, by the way, in our spinal cord. So if you take the possibility of how many different connections there could be, in which way the action potential could travel, taking different routes via the synapses, this calculates to be a number that is bigger than the known stars in the universe. Not in our galaxy, whatever star that we can of. The number of possible routes that can happen in your nervous system is calculated to be greater than that. So how can all this regulate that one single action potential that will travel along the motoneuron? This is what I would like to emphasize by saying that these many synapses here are all electrotonic potential changes and causing local depolarizations and hyperpolarizations, which then add up. And finally, the neuron will decide by adding all these local depolarizations. I wanted to make these yellow and these ones white to show these ones are excitatory, these ones are inhibitory synapses. They all happen to be in large numbers on a single neuron to initiate the action potential. So there's always a large number of synapses on a single neuronal cell, and these electrotonic potential changes between and the addition of these will decide what happens on the neuron. And these receptors, ladies and gentlemen, due to expressional patterns and so on, influences drugs that you add or medications that you add can change their numbers. This is why if you start adding medication, let's say painkiller, after a while it is not enough. You have to increase the dose. Then you have to switch to another painkiller because the one even in a high dose doesn't work, and so on. This is because these receptors can change. So synapses integrate, and this is just a crude and small list of possibilities of how synapses can influence of what comes in and what goes out. And this is not something that you need to learn. I just listed a few that you can, for you to see that even a single synapse can change how it works in time. I decided to select two simple things, a spatial and a temporal so-called summation of responses. The temporal summation belongs to the monosynaptic. The spatial summation belongs to the polysynaptic place. It's very easy to understand. You have a neuron here, two inputs. If you have an input on one and an input on another one, here and there, you see these postsynaptic responses. But if these two give it at the same time, then the postsynaptic response, that is the depolarization here on this cell, will be greater. If you do two at the same point, 1, 1, 2, on top of one another, they can again add up because electrotonic potential changes have the capability of adding together. This is again a small list of possible neurotransmitters that are present in our body. The number now exceeds by far 100. So these are the so-called small molecule, fastly acting neurotransmitters. Acetylcholine is a class of its own. Norepinephrine that I have been describing, dopamine and serotonin that I have mentioned, belong to the amines. There are amino acid neurotransmitters, glutamate being one of the most important excitatory neurotransmitter in the central nervous system, whereas GABA and glycine are neurotransmitters in the inhibitory synapses. Slowly acting neurotransmitters are neurotransmitters that are peptides, different peptides, and we believe that the presence of these peptides is essential and important in memory and learning. How that works and how synapses and neurotransmitters are involved in memory and learning, that's part of the neurobiology studies in the next semester. So, in the last remaining 15 or so minutes, I would like to spend a few words on discussing one more issue, which again relates to electrophonic potential changes. So far, I always said that the action potential is initiated by depolarization that reaches and exceeds the threshold. And of course, if there is a neuron and an action potential comes in the synapse, it can, through this synaptic transmission that we have just talked about, initiate another action potential in another neuron. But the obvious question is, what initiates the first action potentials? Somehow, somewhere, it must be initiated in order for them to run and do what it does. Well, ladies and gentlemen, what makes that? Where does that come from? So the answer, ladies and gentlemen, is that we have cells that are specialized to change extracellular and extra-environmental changes into voltage changes. And these cells are called as receptors. Many receptors you can name since you are in kindergarten. Everyone knows that we have receptors for light, and vision, and our ears to detect sound, to detect touch, to detect smell, odors, and to detect different tastes in the food. But there are other receptors that we have in our body. Our body are full of receptors that detect the blood pressure, that detect where your body in space is. Sorry? I don't understand. I can't hear what you are saying. So, anyone can repeat the simple experiment after me. If you close your eye, you can touch your nose. This is because there are receptors in your joints to detect and tell you the exact position of your body, even though this is not as conscious as vision and so on. And there are receptors that tell you of stretch in your guts and so on. So these receptors, whatever they might be, chemical, mechanical, thermal, heat and cold, up to electromagnetic radiation, this is the light. Even electric fields are detected by some animals. Sharks, for example, use electric fields. And then these physical stimuli will then be detected by certain specialized cells, where these specialized cells will actually initiate a change in the membrane potential of this cell. This will be called as receptor potential. And this receptor potential will then be transmitted towards the central nervous system for decoding. receptors are grouped based on the fact whether this receptor cell is a cell on which action potentials can be triggered. In that case, they are called primary receptors. Primary receptors would be receptors that sense, for example, pain or touch. If the cell cannot generate an action potential, rather it connects to a cell which then can generate an action potential, we then talk about secondary receptors. Okay? First cell in the process generates only a receptor potential. Second cell in the process will generate an action potential. We call it secondary receptor. If the first and second cell does not generate an action potential, but the third cell generates an action potential, we call it tertiary receptor. If the first, second, and third cell does not generate an action potential, only the fourth does, then we would call it, we wouldn't, because that doesn't exist in our body. If it would, that's how we would call it, but it doesn't exist, and there's only one receptor which is tertiary. That is our cones and rods. The eye, as a detector of light, is a tertiary receptor. Smell and taste and many others are secondary receptors. Okay? Mechanical and thermal stimuli are usually primary receptors. Chemicals are secondary. Light is tertiary. Of course, please note that these cells would form synapses with the next cell in line. So what I have said about synaptic transmission is true for these receptor cells as well. So not only neurons form synapses, but receptor cells form synapses as well. They have neurotransmitters and so on. So how the system works is very simple. You have this physical or chemical stimuli, which is then sensed by a molecule or a structure, and then this is then transmitted to an ion channel. And this ion channel will change its conformation and will either go from an open to a closed or from a closed to an open state. I will say an example for both. In your eyes, at dark, these channels are open, and when light comes, these channels close. In your ears, to detect sound, these channels are closed, and when the sound comes, they open, and this is how they generate a change in transmembrane current. They generate a local gradient potential change, again, electrotonic potential change, and this electrotonic potential change is a receptor potential. This can be very easily understood why our receptors must have electrotonic potential changes and not action potentials to start with. Let me give you a simple example. You walk on the street and someone walks up to you and strokes your face. Or you propose something to a lady and the lady strokes your face. And then you have the following decision. Was this this or this? Now you have to decide between the two because your next reaction must depend on you being able to decide whether this was a smoothing action or a slap in the face. Now, if action potentials, since they are all or nothing, and only action potentials would be, you wouldn't be able to distinguish between the two. So receptors must be able to grade the response, and that's what electrotronic potentials do. They grade the response. However, the question then comes that in the central nervous system, where only action potentials are, how can you grade the response? Well, the answer lies in the fact that action potentials have refractory periods. Let's say this here is a receptor potential. If the receptor potential is too small, of course, no action potential is generated. If the receptor potential is big enough to overcome the threshold, then you get an action potential. If this electrotronic potential change is kept on and on, then a second action potential is generated, and a third, and a fourth. The question then is, when is the second action potential is generated? Due to the fact that there is an absolute refractory period after the action potential, obviously some time must be spent before the next action potential is generated. But then a relative refractory period comes, and the strength of the stimulus will decide how early in this relative refractory period can you initiate the next action potential. Therefore, the intensity of the stimulus is encoded in the frequency of the action potential train that it generates. Small stimulus, infrequent action potentials. Large stimulus, big electrotronic potential or receptor potential change, large frequency of action potentials. Okay? Clear to everyone? So this, ladies and gentlemen, is how action potentials in us are generated. One can actually make a correlation between the stimulus and the response that we see, and find the fact that this relationship is not linear. Rather, this is the same graph with linear scales, stimulus, amplitude, response in percent. This is a logarithmic scale, both, and then it becomes linear. So the response is a power function of the stimulus, like that. Please, you do not need to know these numbers. Only the fact that this is not linear, rather a power function. It is important from this that close to the threshold, the change in frequency is the largest, so you can very clearly distinguish between small changes and less clearly differentiate between big changes. This is very important because, let me take my usual example with the lion. If the lion roars, it doesn't matter whether it's a kilometer or 2 kilometers away. But if it is sneaking up on you, it really does matter whether it's 10 meters or only 2. So tiny things are very important to be distinguished. Big things are not that important and never were. So this concludes what I wanted to say about synaptic transmission. I would like to emphasize again that I have done during my lecture that these neurotransmitters, and the agonists are a must. Thank you very much.